Well, we might as well start now. So, this topic is short on detail, but it's an important up-and-coming topic. Basically, the basics of immunology that you learned in the PhD block, where you learned about innate immunity, adaptive immunity, antigen presentation on dendritic cells, activating T cells, producing cytokines, generating inflammation, etc., etc. You considered that as a separate system within the body, i.e. the immune system. But in the past 20 years or more, it has become apparent that the immune system, just like the respiratory digestive system, is an integral part of physiological responses controlled by the brain. The reason why classical immunologists were skeptical about this idea was based on the fact of the so-called blood-brain barrier, that the brain is a completely compartmentalized, independent part from the rest of the body. But with increasing techniques in molecular biology, we've been able to identify gene expression, protein expression, and so on. It's become apparent that this blood-brain barrier isn't as complete as what we've always considered it to be. So the objectives of this lecture is firstly to introduce this so-called neuroimmune network, this crosstalk between the central nervous system and the peripheral immune system. And one of the first types of networks which was identified was the so-called inflammatory reflex, this network which causes you to have high temperatures, i.e. fever, during infections. And then to reevaluate this concept of the blood-brain barrier as an absolute physical barrier preventing influx of circulating peripheral mediators into the parenchyma of the brain. And then to describe, in particular, the microglial cell, which is essentially a brain macrophage involved with multiple functions in the brain with regard to repair as well as brain development. And then finally to describe two types of diseases as a result of inflammatory responses, i.e. meningitis and encephalitis. So firstly, in the very early days, clinicians and psychiatrists had anecdotal data to show that people who suffered from mental disorders like psychosis, schizophrenia, when immunologists looked at their blood cell functions, they found that they had altered immune responses. That if they looked at proliferation or cytokine production from the blood of these patients, they were abnormal. They were reduced compared to normal, healthy people. Alternatively, people who suffered from chronic inflammatory diseases like multiple sclerosis, arthritis, they were often associated with psychological problems as well. Then further experimental evidence looking at gene expression, protein translation, as well as through behavioral psychiatrists, as well as experiments done on laboratory mice inducing them to undergo stress, found that various procedures such as electrostimulation, cortical ablation, could have effects on secondary lymphoid tissue. So, for instance, in mice subjected to stress conditions, such as being housed in environments where there was raised temperature or noxious gases, when they looked at their lymphoid organs, they were found to be reduced in size or degraded. Similarly, then also looking at the transcriptional profiles of various immune cells, such as macrophages, you can find receptors for neuropeptides and neurotransmitters like serotonin, acetylcholine, and so on. Not only that, not only do they express receptors for these neuropeptides, but they can also make neuropeptides themselves. A classical example is the uniform marker for natural killer cells is CD56. This is how we identify NK cells using flow cytometry. CD56 is also called NCAM, or neural cell adhesion molecule, because this surface protein was first identified in the brain, which is an adhesion molecule to allow for brain cells to attach to each other. Further research has shown that CD56 is not only active in the brain, but is also an important cell adhesion molecule for NK cells to attach to their targets prior to killing by injecting perforin and granzyme into these target cells. Another important concept is that within the brain parenchyma, you can find PRRs, you can find HLA expression, particularly in the early stages of development. Now, HLA, if you remember from POD, its primary function is presentation of antigenic peptides. But if there are no T cells in the brain, what is the function of this HLA? So suggesting that HLA has properties other than antigen presentation. We also find complement receptors in neural cells, but we still have no clear understanding what is the role of these receptors. So this neuroimmune circuit is what causes you to have fever when you have an infection. And this is due to crosstalk between the inflammatory cytokines like IL-1, IL-6, and TNF-alpha released by activated T helper cells or macrophages in the event of infection. And these can be transmitted to intermediary nerve circuits, particularly the vagus nerve, and work these signals to the brain to activate the brain to release hormones like CRF, which can act on the hypothalamus to raise the body temperature, activate the hypothalamus to produce CRF, which can then act on the pituitary axis, and then the adrenal cortex to then produce glucocorticoids and corticosteroids to dampen down the immune response. The brain as a result, alternatively, any types of stimuli to the brain can cause the brain to release neurotransmitters and neuropeptides, which can work their way directly to the immune system to either cause immunosuppression or immunostimulation. Central to this type of sensitivity to any environmental noxious stimuli are these pain receptors, predominantly on the skin, called nociceptors, which I think you've covered. Right. So the nerve ganglia innervating the skin are covered with this huge network of nociceptors, which causes, for instance, pain if you cut the skin, or if you exposed to capsicum or capsaicin on chilies, that causes the tingling on the skin. And as a result of activation of these nociceptors, they will induce neurotransmission, resulting in the classical symptoms of the pain response. They will induce CGRP, substance P, somatostatin, which can, when secreted, can go back to have modulatory responses to inflammatory immune cells. Alternatively, these terminals, these sensory terminals, have receptors for cytokines like IL-1, TNF-alpha, IL-6, such that if you have an inflammatory response in the tissue, the release of these cytokines can stimulate the nerve ganglia and then induce calcium phosphorylation and activation, also resulting in this pathway of the releasing of these dampening neuropeptides to dampen down the strength of that response. The nociceptors, based on experiments on mice, one experimental model is to induce skin inflammation by painting on the skin a chemical called IMQ. This aggravates the nerves and activates, and because keratinocytes have a PRR for this, have a PRR TLR7, this chemical produces a pump which activates PRR7. And as a result of PRR7 activation, it causes the production of IL-23. IL-23 is a precursor to induce cells like macrophages or T helper cells to produce IL-17 and stimulate the generation of Th17 cells, which, if you remember, will induce and activate neutrophils. So basically, the upshot is by activating this PRR TLR7, you will induce a Th17 neutrophil type inflammatory response. Another thing with regard to these PRRs expressed on these sensory neurons comes from another model where they deliberately infected the skin of mice with Candida. The Candida yeast has a surface sugar molecule, which is a pump recognized by a PRR called dectin-1. Dectin-1 is expressed on macrophages, but it's also expressed on these sensory neurons. So as a result of a localized Candida infection on the skin, the polysaccharide pump on the Candida yeast is recognized by dectin-1 on these sensory neurons, and this will induce the nearby phagocytic cells to again release IL-23 to activate a Th17 neutrophil inflammatory type response. But in the meanwhile, because this is a network of nerves all connecting to the skin, as a result of this localized activation of dectin-1 at the one site, the signal can be transmitted to other parts of the skin not infected with the yeast, inducing the production of IL-23 and putting those parts of the skin into a pre-inflammatory state to prime these non-infected parts to immediately react if that infection spreads. The vagus nerve, you know more about it than I do, but it serves mainly the gut, right, the digestive system. So it's an important nerve for controlling hunger, blood pressure, as many other functions. But the vagus nerve is also an important checkpoint for dampening the immune response, because any inflammation in the bronchial tissue or the mucosal tissue will signal the vagus nerve to produce these signals through that neuroimmune network to cause the classical sickness behavior you experience, like headaches, fever, tiredness, etc. But not only that, signals coming from the brain as a result of stimulation of the vagus nerve will cause the release of dampening neurotransmission in terms of prostaglandins, etc., to the secondary lymphoid organs to block excessive inflammation. Because if that is broken, then you can get what we call a cytokine storm, where too many inflammatory cytokines are made, which can cause blood coagulation, organ failure, and so on. So the vagus nerve is an important sensory regulator for limiting the amount of inflammation, but also causing the classical symptoms of inflammation in order to help your body develop a good physiological response to the infection. So as I said in the beginning, the blood-brain barrier was always regarded as the reason why large number of immunologists couldn't believe that the immune system was controlled by the brain. And this is based on the fact that the earliest experiments where you injected a dye into the bloodstream of mice, you found that this dye circulated through all parts of the body except for the brain. And anatomists looking at this blood-brain barrier found that this physical junction was due to tight junctions between the cells in the membrane surrounding the brain parenchyma. There was reduced number of vesicles taking in outside fluid into these epithelial cells, and also reduced caniculi and fenestrations going into the brain parenchyma. However, we know that in experiments where we can inject potent pumps like LPS into the bloodstream of mice, the LPS can get into the brain parenchyma, and this is through a directed transport of the pump via the blood-brain barrier into the brain parenchyma via transcytosis through a protein called substance P. We anatomists have also found that there are potential sites in the structure of the brain where there is potential mixing between the peripheral lymphatic system and the brain. One of them is the choroid plexus, whereby plasma from the lymphatics mixes to produce CSF. The other is that the brain, being well networked with blood vessels, in the perivascular spaces there are macrophages, dendritic cells, T lymphocytes in those sheaths, which have the potential to exit into the parenchyma. The other thing too is that the brain being enclosed by the skull being bone also contains bone marrow, which has stem cells which potentially can develop and then house or get into the parenchyma. About 10 years ago, it was discovered that in the brain structure, there is a network of lymph nodes. But this is still undergoing a lot of research because it doesn't. So the CSF is there, similar to what the lymph does of removing waste products from the brain and recycling them. If there is a lymphatic system in the brain, you would expect that it would act just like secondary lymph nodes, such that any antigens processed would be taken into these nearby lymph nodes for presentation to T cells. But that system doesn't seem to operate there. So we still don't really understand how this lymphatic system within the brain works. Within the meninges, so if this is the bone of the skull, and then this is the meninges above the the pia mater, you have a full complement of immune cells: T helper cells, dendritic cells, NK cells, mast cells, all of them to induce inflammation, classical inflammation, in the event of meningitis, such as infection of the meninges. The progression of that disease, these inflammatory cytokines could penetrate this membrane to get into the parenchyma, where it can then cause more serious defects. So then, as I said, the inflammatory cytokines like LPS, pumps like LPS, as well as inflammatory cytokines, it's been found that they can migrate through the meningeal membrane to get into the parenchyma through transcytosis mediated by this protein, glycoprotein P, which can transport the cytokines into the brain parenchyma. But you don't get these cytokines moving in the other direction. So even though we know that there is no large infusion of inflammatory cytokines into the brain, it seems that there is regulated, that there is possibility for these cytokines to get in through regulated movement. But exactly what they do, the effects of these cytokines on the brain parenchyma, we're still not clear about. One of the classical models for multiple sclerosis is called experimental autoimmune encephalomyelitis, where they inject myelin basic protein, which is the predominant antigen for multiple sclerosis, into the brains of mice. And what they find is once they inject this protein, they get appearance of interferon gamma in the CNS. What is the source of this interferon gamma? It's not clear. Maybe it's microglial cells, or it's maybe these accessory inflammatory cells. Another thing that's been found is that in experiments where they've deliberately broken the spines of mice, they find that in the spinal column, IL-4 is being produced. And this IL-4 has the ability to repair neural connections. So a lot of research is now being going on experimentally to see whether we can connect severed nerves using various cytokines. So the basic message from this is that there's a degree of cytokine stimulation in the brain. And these can either be destructive or regenerative, depending on what cytokines are available, how much they are, the timing, and so on. With regard to pathology, there are two types of diseases in the brain parenchyma, which is either neurodegenerative, as in the case of Alzheimer's disease, where the current idea is that activation of microglial cells results in the production of cytokines like IL-1, TNF-alpha, which can affect the axons, causing destruction. With neuroinflammation, either through meningitis with inflammatory cells penetrating the brain parenchyma, or in the case of multiple sclerosis, where T helper cells infiltrating the brain can activate B cells to make autoantibodies against the myelin sheaths. That is what seems to be the models from current experimental data on mice. Again, all the classical cytokines that you've gotten to know and love for stimulating IgG production and so on. So the microglial cells we accept originate from the macrophage, the monocyte macrophage lineage. They express TLRs like TLR4 recognizing LPS, just like peripheral macrophages. They, as well as playing a protective immune role, they also have an important function in maintaining homeostasis of a healthy brain, of removing dead nerve junctions, nerve fibers, as well as repair. Based on increasing evidence for this crosstalk between the brain and the activity of the nervous system, clinical trials are underway, such as things like behavioral therapy, whereby improving the psychology of a patient with a chronic disease like terminal cancer or whatever, the belief that if you can increase the mindset of the person, you can increase the activity of the immune response to deal with the cancer infection. The other is to use external influences like pharmacological treatment or electrostimulation to stimulate parts of the brain, thinking that you might be able to improve or stimulate particular immune responses like cytokine production or antibody production and so on. So, in conclusion, consider the immune system as a normal physiological part of the body controlled by the brain, which is not a separate system, and that even though the blood-brain barrier is a strong physical barrier preventing circulation from the lymphatics, there is still parts like the choroid plexus where you can get infiltration of cytokines as well as immune cells into the brain, and that there are cytokines which can have effects on neural connections, which might even have an effect on things like memory and learning as a result of cytokines. Okay? So this seems like an abstract type of lecture, and every year I have problems with providing questions for your assessment. I only have to provide 4 or 5 questions. So my answer is the questions that I ask are more directed towards your basic immunology, i.e. inflammatory cytokines, blah, blah, blah. Don't worry too much about the neuro part of it, okay?